now we have our final speaker of the day coming to us from the FDA. So Dr. Michael Pacanowski is the Associate Director for Genomics and Targeted Therapy in the Office of Clinical Pharmacology at the U.S. Food and Drug Administration. His team of translational scientists is charged with advancing the use of genomic and other biomarker innovations to maximize individualization in drug development. And to that end, Dr. Pacanowski oversees a program focused on reviewing investigational new drugs, developing policies and processes, engaging stakeholders, and conducting regulatory science research. Dr. Pacanowski received his PharmD from the Philadelphia College of Pharmacy, a Master of Public Health from the University of Florida, and also completed a residency in clinical pharmacology at Bassett Healthcare in Cooperstown, New York, and a clinical research fellowship in cardiovascular pharmacogenomics at the University of Florida. Please join me in welcoming Dr. Pacanowski.
Thanks for the introduction and having me. It's my first time in Minnesota, so it's really a pleasure to be here. Do appreciate the wonderful meeting that's been put together so far. I think it's been very enlightening, a pleasure to hear all the different talks, and I'm really impressed that we still have a pretty good crowd here down to the end of the wire. So thank you for that. So what I'll talk about is mostly some of the regulatory perspectives on genomics and biomarkers and precision medicine. And being at the end of the day, with the lineup that we had here, I had the good fortune to not include the words abacavir, clopidogrel, warfarin, and some of the other drugs in any of my slides. Happy to talk about it if you want during the questions, but I'm not going to talk about it during the slides. Rather, I'm going to just give a more higher level orientation to some of the precision medicine initiatives going on at FDA, as well as some of the regulatory processes and principles that are in place. So I'll start talking about biomarkers, precision medicines, and then get into some of the guidance and policies that have been developed over the years, as well as a brief discussion on the Qualification program and precision medicine initiative. So I think most of you are probably very well aware that we have a suffering pipeline. It's been estimated that approximately 1 in 10 drugs make it to market after being studied in first-in-human trials. That's a pretty abysmal business to be in, if you ask most. What's also a little bit disconcerting to me is if you look at the success rate out of phase 2, it's still about 1 in 3, and then the approval likelihood is still only 16%. So there's a major issue there in that we're not perhaps killing enough compounds very early on, or that we're investing a lot in phase 3 trials that are proving ultimately to fail, which is quite a costly venture. Now, if you look at the root causes of this, most of it ends up being on the efficacy side of things. Drugs either don't demonstrate efficacy or they fail to differentiate from other products in the marketplace. And that's the major driver for attrition in the pharmaceutical pipeline. Now, many had hoped that genetics and biomarkers would have been sort of this technology that would save the pipeline. It would allow for smaller trials. They could be done faster. We could get drugs to market quicker. And I think by and large, that may be true. We've had a number of impressive examples in the setting of oncology and some rare diseases, which have really shown us that this is a viable strategy to bring drugs to market. But it doesn't necessarily mean that the drug development process becomes less complex. You end up dealing with 2 different regulatory infrastructures, both on the device side as well as the drug side. So it does create for a complicated drug development program trying to move 2 products at once forward to market at the same time. But nonetheless, this promise of quick wins and fast fails is something that ultimately is viewed as being desirable for the pipeline. This has been borne out in a number of analyses. A lot of companies have deconstructed some of the successes and failures in their pharmaceutical pipelines and essentially showed that there's a mix of key things that we often probably in this room might take for granted. Basically, knowing what your drug does, where it goes to in the body, how to dose it, who to give it to, seem like very basic things. But unfortunately, that's not necessarily the case for all the drugs that come to market. There's often incomplete information on a drug's mechanism or potentially what vulnerable populations require a reduced dose, for example. So, in this particular case, looking at the pipeline, they found that if they understood really well the bioavailability of their product, that they had biomarkers that were able to show proof of concept and show that the drug was active, that would help them with dosing. All of these types of things actually ended up promoting success of products in their pipeline. So it's become an operating philosophy of some companies to have a lot of this information built in very early on. So I use the term biomarkers and use it very broadly. And biomarkers and precision medicine are really kind of the basic, biomarkers are really the basic element of precision medicine, at least as we view it in the modern sense of the term. One thing I'd like to point out to you, and I think it's been discussed earlier, is language and communication is of the utmost importance. Because of this, FDA and NIH over the past year and a half have been meeting regularly to try and develop a basic taxonomy of different terms in the field of biomarkers. So we published this resource on the NLM website. You can go visit it. It essentially defines biomarker, as well as multiple different types of biomarkers that are the different categories and what they inform you from a health standpoint. And basically what we intend to do is update this over time, so it'll be a living resource. If you have any feedback or input on it, feel free to use the email at the bottom of the slide here to send your input. But one thing I'll point out is a biomarker is really not an assessment of how a patient feels, functions, or survives. That's really the basic premise of what a biomarker is. And the various categories could range from a diagnostic biomarker, which is used to confirm the presence of disease, a monitoring biomarker that's used to evaluate changes in a disease or survey for disease that might be emerging. They could be predictive of drug response. They could inform an individual's susceptibility or risk for a disease. So these are the basic flavors of biomarkers, and they're spelled out more clearly on the website. Now I give this as sort of prologue because I think it's important that we all have the same basic lexicon in trying to talk about this field. So now I'll get into precision medicine, which is more in line with what we're talking about here today. Everybody's pretty well aware of the Precision Medicine Initiative at this point. I have the obligatory Obama slide. So personalized medicine, as it was often called, was really medicine. It's how far a patient has to drive to the clinic, how much money they have, how much they can afford for a prescription, all of these basic things that make up basic medical practice. So precision medicine's become the term of art, and it's really, I think, built on a foundation of molecular testing and using some of these new technologies to help understand what disease looks like in an individual patient to help make better prescribing decisions. Pharmacogenetics, for example. And we've had a number of cases over the years that have been very illustrative of how this actually plays out. So precision medicines, the way I generally use this term, really are drugs or biologics that are intended for use in a specific genetic subset of a disease or other biomarker-defined subset. And it's used basically to identify the dose, select the patients for a treatment, or to monitor their response to treatment. In the drug development context, often the drugs have a diagnostic or prognostic value when they're used as part of clinical trials. More and more, we're starting to see programs that are co-developing predictive biomarkers, and these are things that are generally related to the pharmacology of the drug and are putatively identifying who responds to the medication. Now, the way we get to this point in a drug development program could be a number of different pathways. It could be a mechanistic relationship, pure mechanism, i.e. you don't express a drug target, you don't enroll that population in clinical trials. Or you might have some early preliminary evidence that would show that the drug actually has some differential activity based on the presence or absence of a biomarker, and then that would carry forward into a targeted or precision drug development program. So the way this actually plays out from a clinical trial standpoint is pretty straightforward. You can either enrich your clinical trial for those patients who have whatever molecular profile you're interested in, or you can stratify your clinical trial. Basically enroll all comers and either stratify randomization up front or do retrospective analysis when the trial is completed. And that's the basic gist of it. And the outcome of this is actually pretty simple as well. You can either select doses, select patients, or monitor response, as I mentioned before. So there's not really many variations on this theme. It's relatively simple in the drug development context. Now, I mentioned before that this drug development and biomarker development, when it happens simultaneously, can be a pretty complicated venture. Because basically what you have is you might have the biopharma industry developing biomarkers, running tests on their early clinical trial populations to see if there might be a certain subset of patients that responds well or doesn't respond at all. Once something is identified, the pharmaceutical sponsor has to then commit to this targeted development strategy. And in doing this, they may or may not have to bring a test to market. So oftentimes, they'll have to partner with a diagnostics firm and then coordinate the development of the two products as they start to conduct their late phase clinical trials. Some pharmaceutical companies have in-house capacity to develop the diagnostics, so they do actually have their own divisions related to diagnostics. But then the ultimate goal is then both the drug developer and the test developer bring submissions for marketing approval together at the same time. They get approved, and our experience to date has suggested that oftentimes this happens. We have the approval coordinated, so it happens on the same day. So going back a little bit to talk about some of the transition points in this targeted drug development enterprise, ideally it's something that you want to plan from the outset. I mean, it's very easy when you have very robust biology, a very deep understanding of the molecular pathology of a disease, that you can take from your very earliest trials a very targeted subset of patients, get to proof of concept that the drug is having some activity very early on, and then use that as a springboard to conduct the dose-ranging trials and then clinical efficacy studies. So this has been pretty much the more common type of scenario in the cancer setting. Outside of cancer, clearly we don't have as deep of a molecular understanding of disease as we'd like. Common chronic diseases are complex from a genetic standpoint. There's a lot of environmental factors. So often what we end up seeing outside of oncology is these exploratory studies in phase 2 that might identify a biomarker that could be predictive, and then they carry that forward to the next phase of development to confirm whether the biomarker actually does indeed predict response. So that's the more typical flavor outside of oncology. In rare exceptions, we identify some things in the late phase or post-approval settings even. A lot of the biomarkers that were identified and discussed today were all pretty much identified in the post-marketing setting. And we're now seeing a shift actually for a lot of this science to becoming earlier in drug development. So you see a lot of newer drugs being approved with labeling language related to dosing and whatnot, rather than this cropping up in the post-market setting. Again, this can be, from an evidentiary standpoint, built on mechanistic hypothesis. You might have preliminary evidence based on clinical studies, or there could be a risk-benefit profile of a drug product that might push you towards a more targeted strategy because the toxicity profile is unpalatable, but the drug may have beneficial effects. And that may be leading one to pursue a more targeted development strategy in order to get the drug to market. So this is a slide, forgive the brand names here, but just goes to show you the number of drugs that have been approved over the past couple of years that have some element of molecular testing associated with them. Obviously, a lot of these are in the setting of oncology, but clearly hepatitis C's undergone a revolution in the past decade, largely built on our ability to sequence that genome. And we've got drugs for cystic fibrosis that now have the potential to be disease-modifying rather than just treating symptoms. So I think a lot of this genomic revolution, while we're struggling with all of these post-marketing clinical implementation approaches, is really proving to be quite successful from a drug development standpoint. Now, clearly it's not pan pipeline, but nonetheless, we are getting a lot more experience. So in deconstructing some of these cases, these approvals that we've had over the past couple of years, you see that a few things start to emerge, a few common themes. The nature of the disease. Clearly, if it's a serious and life-threatening disease, we have an interest in trying to get drugs to market. If a biomarker is the tool to do that, then that's what we end up pursuing. And in some cases, efficacy has been demonstrated in a very small subset, even though the drug could be effective in a more broad population. It's been developed for that subset, and there have been post-marketing studies to look at it more broadly. In addition, the risks and benefits of the drug product weigh heavily into these precision medicine programs. Basically, if the safety profile of the drug is not superb, then again, there's a likely interest in shifting that risk-benefit profile by enrolling only those patients that respond. And then the biomarker also has its own mix of merits and limitations. Clearly, biomarkers that are well established in practice are very easy, much easier to build a program off of 17p deletions in leukemia, for example, and lymphoma, or leukemia, are really emerged as a prognostic factor for outcome. Patients with 17p deletions fare rather poorly relative to the rest of the population with the disease. And as a consequence, there were some drugs that were developed specifically for that subset and were approved in the first-line setting for them. So clearly there's quite a mix of different things that weigh into some of the decision-making on whether or not it's appropriate to pursue one of these strategies. So I mentioned that there's test development that co-occurs with these drug development programs. This is a snapshot, fairly old at this point. There have been a few others, but the Center for Devices and Radiological Health has approved a number of companion diagnostics. And just by way of definition, a companion diagnostic is a test that's essential for safe and effective use of a therapeutic product. And those are essentially listed out here. Again, most of these were approved on the same day as the drug product. So the FDA labeling often, or the drug product labeling often comes up as part of these discussions in pharmacogenetics. Our most recent look at the landscape shows that there's almost 200 gene-drug pairs that are described in labeling. But I'd like to just reiterate one point that I think is often overlooked. There's not 200 drug-gene pairs that have prescribing recommendations based on a genetic factor. It's actually really only about half of those. We often put genetic information in labeling because it's a feature of a clinical trial. For example, a drug interaction study might exclude poor metabolizers, so we include that, although it has no effect necessarily on the drug of interest per se. So just something to think about if you ever go to this table and reference it, read the prologue, and it sort of tells you exactly what's in there. So it's not all of the drugs that necessarily have pharmacogenetic prescribing recommendations. But nonetheless, about half of them are. That's quite a fair amount. So there's quite a bit of drugs that do indeed have prescribing recommendations based on genetic characteristics. So I'm going to switch gears for a couple minutes and talk a bit about the regulatory framework. So there's a couple of different pathways by which a biomarker can come to be used as part of a drug development program. So one way is through the investigational new drug review process. I've talked a bit about that. All of the examples that you've seen were essentially developed by an individual pharmaceutical sponsor, test developer, working in a coordinated fashion and consulting with FDA on the progress of their program. The other program that we have, just that I wanted to make you aware of, is the Biomarker Qualification program. This is a program that is trying to foster development of biomarkers that could be used very broadly across all drug development programs, for example. Some cases that we have in development are biomarkers of renal injury, for example, by the Critical Path Institute. These are indicators of nephrotoxicity that could be used as a tool as part of drug development to detect whether or not kidney injury is occurring with a molecule. So this program is intended to be very broad, so it could be used for issues, biomarkers that might be useful as enrichment factors because they're for prognosis, diagnostic biomarkers, safety biomarkers, and the like. So most of what I talk about is generally related to the investigational new drug review pathway. And looking back at some of the examples, our regulatory framework as it stands today is built on experience, as I've mentioned. So I think we've all learned and appreciated with the examples built on less than perfect retrospective evidence end up being very challenging to translate into practice. And when you take an approved product and try and put a biomarker on it and change the way a drug is used and prescribed, that's a very challenging proposition. Warfarin, everybody measured INR for a very long time. It's very difficult to put a new test on that because we've already changed the prescribing paradigm to allow for the variability and monitor INR. So this came to a head, I think, really with KRAS and some of the anti-EGFR therapies. There were a number of studies that showed that KRAS mutations altered the response to two anti-EGFR monoclonal antibodies. And this was challenging because the clinical trials didn't necessarily collect tumor specimens on every patient. It was done retrospectively. There could have been variability in tissue quality or the assay that was conducted. So it presented a lot of challenges. But nonetheless, that was based on a very high degree of biological comfort because we had a long history of understanding how KRAS impacts tumor biology. So there's a really hybrid of evidence that has to come into play when dealing with this, but nonetheless it's difficult when trying to retrofit biomarkers onto an existing product. In addition, we've had some cases where the clinical trial assays that are used as part of the drug development program may have been flawed or not done in a central or high quality way, and that really jeopardized the program because when you have a clinical trial that's conducted and you end up reclassifying 15 or 20% of patients at the end, that statistically is not as robust as it was originally designed. You begin to lose randomization and things like that. So that's another issue that we've had to contend with often in the review of these programs. We also have found in a couple cases that the companion diagnostic co-development strategy, while with the greatest of intentions, produced a drug that could be effective more broadly. We've had cases where the test wasn't viewed as necessary at the end of the drug development process because the drug did something important. But we have now had a couple of cases where we've approved a complementary diagnostic, a relatively new term, that might be informative for using a drug but not necessarily required. And then, with the tumor mutation profiles we're dealing with, as well as like ivacaftor and some of the other rare genetic diseases, clearly not all mutations are created equal, and that's presenting challenges of how to put patients together in clinical trials, something we're very actively thinking about. So with all of this experience, since, for the past decade, we've published a number of guidances. There have been a couple amendments to rules in there, as well as some international guidelines that have been developed. It's probably not the most exciting reading on the planet, but if you ever wanted to go dig deeper, you can find these on the FDA website. I'll highlight some salient points of a couple of them. So we have guidance on clinical pharmacogenomics and early phase studies, and the intent of this guidance is really basically to promote DNA collection in clinical trials. We essentially provide some scenarios where DNA collection would behoove the sponsor. For example, the drug has high variability, they've observed race effects, there's idiosyncratic reactions, and the like. We would actually strongly encourage companies to begin collecting DNA to allow for exploratory analyses. We also say if there are known factors that are likely to influence response, so if you have a genetic susceptibility to thrombosis via factor V Leiden, you've got a drug that increases thrombosis risk, you should be testing for factor V Leiden. That type of scenario is what we try to also reinforce in this guidance. This is now more recently translated to an international guideline. So there's an international conference on harmonization effort to provide sampling recommendations more globally, because as you may know, DNA sampling in different parts of the world can be quite challenging. Some countries have different storage and retention times with samples, there are shipping issues, so there's really a much needed international endorsement of how DNA is collected in clinical trials. So that's something that's being done to really enable a common understanding and state the interests of the different regulatory agencies worldwide. One of the more important guidances in this area is clinical trial enrichment. This basically is a document that outlines sort of FDA's recommendations around clinical trials that are conducted in subsets of populations. Basically, every clinical trial is enriched. It's not the general population that's enrolled. So we provide some parameters around this, and it's particularly important in the precision medicine context because we allow a lot of trials to go forward where only a biomarker positive population is enrolled, and there's a lot of discussion around, well, when do you enroll this marker negative population? Who's in that marker negative population? What kinds of studies need to be conducted? And can they be done pre or post marketing? So that's some of the salient points that are discussed in that guidance. Another one is a rule. Basically, actually I'm going to skip this one. And then the companion diagnostics guidance, as I mentioned before, companion diagnostics again are tests that are essential for safe and effective use. We apply pre-market review and risk-based regulation of these products, and that we basically have an expectation that there's a device that comes to market with the drug product if it's only to be used in a certain subset of patients. I mentioned the drug development tools qualification program. Biomarkers is not the only one. We also have them for clinical outcome assessments like patient-reported outcomes, as well as animal models, in addition to biomarkers, which we've discussed already. So I'll end briefly just by touching on some of the precision medicine initiative activities. This has been mostly led by the Center for Devices. As mentioned before, there's this national cohort, there's a lot of investment in NIH and National Cancer Institute. FDA also had received some funding to help come up with creative strategies around the regulation of next generation sequencing tests, which is something I think we've been hearing quite a lot today. Next generation sequencing obviously is not the same as our traditional testing. It's not a single sheet of paper that comes to you from LabCorp, Quest, or whomever you order your test from. It's more voluminous, obviously, and needs a completely different framework in order to extract information from it. So from the FDA perspective, basically the vision is to implement new policies that would promote research and accelerate the translation of these technologies into treatment benefits. And that means a number of different things. It's in the near term trying to develop standards from an analytical standpoint as well as a clinical validation standpoint for making claims and using next generation sequencing tests, but also developing shared resources that will help enable the development of knowledge and that could be eventually used for patient decision making. In the longer term, again, it's this regulation that's more standards-based, and the bottom line is really to make sure that patients receive accurate, reproducible, meaningful results, and at some level that this is portable across the healthcare system. So there's a bunch of different efforts that fall under this umbrella of the Precision Medicine Initiative, but basically the FDA is working on developing the standards to ensure quality. They've developed open source tools to help the test developers meet these standards. Precision FDA is one pretty successful example that's started just in the past several months. And then also promoting translation through a flexible, dynamic regulatory system. And you'll know we have a couple next generation sequencing platforms that have been cleared by the Center for Devices, so they have undergone the rigor of the analytical process. They still test for a limited number of loci, but nonetheless, we've gone through this and have been thinking about this for several years. So there's a lot of areas that are also on our mind beyond next generation sequencing. We've had a number of workshops and working groups on a variety of different efforts in this space. You'll see a lot of these are very diagnostic centric, but we in FDA also conduct a lot of research, and it's been a challenge to create an informatics architecture for this type of data, but we have actually a pretty impressive high performance computing environment that was developed basically de novo by the Center for Biologics. But we've also tried to understand what the standards are for submitting data to the agency and whatnot, though we have and successfully reviewed it. Panels are obviously of great interest, particularly in the oncology setting. More recently, complementary diagnostics, as well as evidentiary criteria for these biomarkers that can be used more broadly. So those are a variety of areas that are currently in the works. So just to summarize, the success of precision medicine really requires a couple of key things. Tests that are safe and effective. None of us wants a BRCA test result and pursue surgical procedures to remove things based on a test that was faulty. That would be crazy. We want learning health systems that basically allow us to generate new information as it's emerging. There's a lot of discussion on real-world evidence, use of these third-party health systems, electronic medical records, and other things. Integrated framework that would allow for synthesis of information from patient experience would certainly be helpful in this setting, particularly because we're often dealing with Ns of 1, 2, 3 when you get down to rare mutation profiles. Targeted therapies, obviously, that are effective and safe, and some of this at some level depends on having a permissive but but flexible and innovative regulatory and research enterprise that would catalyze the development of these products and deliver them to patients. So with that, tools, innovative drug development programs, solid next generation tests, ultimately, hopefully, will lead to better patient outcomes. And with that, I'll close.